Hi, and welcome back to Bio 131. We're in the second part of our chapter on 27 on prokaryotes, bacteria mostly. And what I've been doing this one, so the last one you should have gone over, because I'm using some of the terms from that, is the structure of the prokaryote. That was in part one. And in particular, we're going to be talking about motility. And so we'll be talking about the flagella and the structure of the flagella. So in this part, I'm going to take up motility. We're going to talk about the DNA present in the cell and where it's present. It's in two parts, something we call a genophore, the single chromosome, and also has plasmids, which are very rare in eukaryotes. And then the third part we're going to talk about is how they reproduce, and they reproduce by a cell division called binary fission. So let's get started. Okay, and so I'm going over motility, chromosomes, plasmids, cell division, genetic recombination. There's three different ways to do genetic recombination when we get to it. Viewing back up here, we have a view of the structure. We have three basic cell shapes in the bacteria. Anarchy, and that would be round, the coccus, the bacillus, and the spiral. And what I ended with the last lecture talking on was the flagella. And I mentioned how it's solid protein, that it rotates like a corkscrew or propeller, and it has a motor at the base. And it's the only known true rotor structure that we know of in cell biology, where actually is an internal tube spinning or being pushed around by an outer ring. It's kind of very interesting. And that's called the basal apparatus. And here's the structure of it. So you can see this part to the outside from the hook, and the long extension is the filament. Right, that's rigid, so that's made once and it extends out. But what's propelling it is an electron transfer and a proton gradient that's taking place between these membranes. Looking at this one, looking at the cell here, we notice there's an outer membrane, a thin peptidoglycan cell wall, and an inner cell membrane. So take a minute and think about, is that gram positive or gram negative? And so this is showing you an example of a gram negative. I mentioned how the flagellum is made up of 21 separate proteins. You don't need to know that. It's just that so if we think in evolution, because we have the introduction to evolution chapter, how do things come about? They come from smaller structures, slowly modifying over time and becoming more and more complex. And so it was a very interesting and thorough investigation being made as to how this very complex structure could come about, because if any one part were missing, it wouldn't work. And that's today's structure. But anyway, so they found out this actually based on over 21 separate proteins, and the origins of the different separate proteins were elsewhere in the cell in some places and had simpler features, and they all combined to make this. So there's a whole story there in microevolution about, microbiology, about the evolution of the structure. So you don't need to know that fact, but just kind of think in the back of your mind. So even these very complex structures have simpler origins, right? Again, it's analogous, right, to the eukaryotic flagella. It does not have the same structure. It doesn't have the same mechanism, so they're not homologous structures. Okay, so the bacteria are using these things to move. And so they could just move randomly, and some are in their environment. But what the bacteria have been shown, different ones, to respond to different things in their environment, and either go towards those stimuli or away from them, right? And so this movement in bacteria referred to as taxis, right? And so if they move towards something, it will be positive, right? If they move away, it's negative taxis. And so if it's light, it's phototaxis. If it swims towards the light, it would be positive phototaxis. If it moves away from light, it would be negative phototaxis. Anything dissolved in the environment we call chemo. And so if it can sense that and move towards a sugary solution in the media, right, that would be positive. If it swam away from it, it would be negative chemotaxis. Some bacteria, especially in soil, have the ability to sense magnetic fields, and the ones that have been shown have a positive magnetotaxis that will actually swim and align with the magnetofields, right? We're not exactly sure why, so there must be something with that that's positive and being reinforced by evolution, but at our level of understanding, we can't quite figure out what exactly that is. It may have something to do with mineral uptake, that the more are aligned through there, but we don't know. So I just want you to think for a minute. And so this is only a single cell. It has no eyes or nose or mouth to see the light or to taste or smell the chemical. It has no organ to sense the magnetic fields that maybe birds and some insects have for migration. Right? So how does a single cell function in this way that it could sense and receive the stimulus and then direct its movement? Right? So it's not a whole organism. I mean, it's a whole organism. It's not multicellular. It doesn't have organs. So how does a single cell do this? Right? And so it has receptors on the cell membrane that are either for light or for the chemical or for somehow the magnetic field. Right? And so it has those receptors, but how does it turn it into that movement? It can't think, this light's over here, I'm at point A, and I'm going to swim towards point B. And so what happens is they simply, what are called run and tumble. And so when it's running, it's simply moving in a direction, and then it'll go for a certain amount of time, and then it will just spin around and move in another direction. And so the run is when it's moving along an axis, right? The tumble is simply a spin. And often after the tumble, it will be moving in a different direction. And so if there's no stimulus, it will simply randomly move throughout the media. But if it's directional, and in this case this will be positive, and here's an attractant, so that would be chemotaxis. Simply what happens is that it runs more and it tumbles. So when these receptors are getting a positive stimulus, right, it will allow it to run more. So basically, as long as it's getting a positive stimulus, it turns down or reduces the amount of time it tumbles, right? When it's moving away from the stimulus, it will actually cause it to tumble more frequently. And so overall, you can see it's not this direct line. It's not like it's started at A and went directly to B. What a bacterium is going to do is going to go to A, run, tumble, run, tumble, run, tumble, run, tumble. But it's running more than it's tumbling. So overall, generally, it will get closer to B. And so here, I want you to think about what is the response. Looks like my face is in the way. I'll move myself up here. So here's the start. So this is where the start of the experiment is, and we have some solution in here. Oop. My pen is making it jump ahead. Has some solution in there, right? And so you want to know what kind of response. Is it positive? Is it negative, right? And there's a solution, so that's making a chemotaxis. And so this is when it's starting. So at first, we just have the solution in here. And this is where it's growing. We call that the media. So now you want to see and look at the bacteria. Are they going towards, away, or random? Okay, and so if we look at this first one, you can see the bacteria are actually in a higher concentration now on the inside than outside. So it seemed to me that they're moving towards that substance that's in this little micropipette. And so that would be a positive chemotaxis. This one is negative because they're actually moving away. But notice there's two little ones that got trapped in there. Because remember, run and tumble, run and tumble. So one little one, when it was here, when it first started, that one got tumbled in here. And no matter what it does, it got trapped. So a few of them did get trapped in there, right? But if you look, the vast majority could run and tumble away. So that would be a negative chemotaxis. And here, as many are tumbling in as they're going out, so that would be no chemotaxis. That would be random. So we have a positive chemotaxis, we have random no chemotaxis, and then we have negative chemotaxis, moving away. Okay, next I want to talk a little bit about the different levels of DNA present in a cell. And in prokaryote cells, there are two sources of DNA. All of the essential genes, what makes E. coli E. coli, or what makes pneumonia, right, pneumonia, are in its single chromosome, right? And that's called the genophore. Right? So it's one chromosome, one single chromosome. Right? I'll put that term in here. It's double-stranded circular DNA molecule, right? It's similar to our chromosomes in a little bit in that it has the basic DNA or genes that determine all of the features, right? But what's different is that we have multiple chromosomes. Our DNA is also double-stranded, but they're not in a single little loop, right? So the structure of ours is a little bit more complex, right? A couple things that are important, that during cell reproduction, called binary fission, it will actually attach to the cell membrane in one little bit. It's not like it just lines up and glues the whole thing. One little part of this chromosome, like a little hook, will attach over to the membrane, just at one little area. Right? Something similar we'll get to, and we'll use the term centromere when we get to talk about eukaryotes. But here it's not termed that. But it has the same thing. It's one little part that's going to be used to pull and move the chromosome. Right? There are two terms that are used back and forth, and for us they're kind of synonymous, but they're not. The nucleoid is the region of the cell inside where the single chromosome spends its time. The genophore is the DNA in the chromosome, right? So they're kind of interchangeable. We're talking about the same thing, but one specifically is the DNA, and that would be the genophore, right? And again, it contains all of the essential genes for the cell to survive. So what makes that species? All right? It, prokaryote, bacteria, and archaea, also have a second source of DNA, and these are called plasmids. And so if this is the cell, right, this would be the large single chromosome. It will have much smaller bits of extra DNA, and these will be plasmids. Usually we're always referring to it in plural because there's so many of them. And again, these are extra genes, right? Anything that pneumonia needs to be pneumonia is on its chromosome. That's what makes it that species, right? The extra traits it might have, and the one that we'll be talking most about will be antibiotic resistance, will be on these plasmids. They can be a single enzyme that helps them convert a different type of sugar, right? So it could be a variety, but they're not the key feature, and there's not lots of them. It's one or two individual little traits that can be passed among the different bacteria. And that's the other thing that's different about them. The plasmids can be lost or gained, right, from the cell, where the genophore can't, because the cell would die without it. So they can be transferred. And the partners that can be transferred among don't even have to be as closely related as exchanging the genophore. And so that information when we get to recombination. So that makes these plasmids much more evolutionarily advantageous because they can move around. They don't even have to be the same species. They're not completely unheard of outside of the prokaryotes. For example, in yeasts, which are a eukaryote, a fungus actually, they also have plasmids. But they're really rare, nearly unheard of outside of yeast and eukaryotes. So this is a typical feature of prokaryotes and bacteria in particular. And so here we have the bacteria cell that's been opened, lysed using heat. And you can see all of these lines out here. That is the single folding back and forth loops. That is the single chromosome. And over here, we have plasmids. So you can see they're much smaller. This is probably also a plasmid that got ripped apart. Looking at it, can you find another plasmid in the photo? So if we look up top here, here's another little plasmid not labeled there. So you can see the amount of DNA in the plasmid is tiny compared to the single chromosome. Other thing to remember about it is that they can gain or lose these plasmids. So this one can pick up a plasmid from another cell, and during replication, it might lose, one daughter cell might lose a plasmid. So they vary. But the chromosome itself will remain in each daughter cell. So if we look, this is just showing you the amount of DNA present. And let's see if I get myself out of there somehow. I'll put myself over there. And so let's say, obviously, vertebrates like us with multiple, 46 in our case, chromosomes, have notice it's also an order of magnitude down here. All right, so we have 1 million times more DNA than a single prokaryote. Right, so they have a small amount single of DNA, so they don't carry as many genes as we do. But when we get to metabolism next time, as a whole group, the bacteria are much more diverse than the eukaryotes. Okay, so they grow by a process called binary fission. And so I'm going to draw a cell dividing, and so see if I can start this up correctly. Black pen, so I'm going to put cell wall here. Draw some irregular, oh, still see I'm going to make a loop out of it. I need to turn that off. Okay. Inside it, I'm going to put the plasma membrane. I'll use both terms. I don't know why there's two terms. It's interchangeable. Plasma membrane, cell membrane, they mean the same thing. Rigid cell wall to the outside. And what color DNA do we want? Let's do orange DNA. And so it's going to have a single loop chromosome, and I'm going to show you how it's coming and attaching to the cell membrane. And then using a process very, very similar to the process that happens in eukaryote cells like yours, the DNA will start replicating itself, actually in several spots. So you end up with an exact copy of the DNA inside the cell. And so it replicates, it copies itself. And then what happens is the cell kind of elongates a little bit, still attached to the cell membrane. But now these loops of DNA will start separating. I say pull because what's actually happening here is the cell membrane itself is lengthening as the cell lengthens, and their attachment points are separate, so it's actually growing membrane, new membrane between the two parts. And then our cell gets a little bit larger. Membranes is formed, and now the single loop DNA are separate. And it starts forming in new cell wall until it folds across with new membrane. And then we end up with two daughter cells with their chromosomes. And this is called binary fission. It's supposed to be a B there. And that simply means splitting in two. I'm going to go back up a little bit now, and I'm going to add into our original cell two plasmids. And there's multiple copies. So I'm going to do a blue one. And what color haven't I used? Are there more colors? And I'll do an orange plasmid. I'll do green, we'll use green. And a green plasmid. So if we go down here when it's dividing, we have the green plasmid, and we have 3 copies of the blue plasmid. And then you can actually see what's happening here, and what I'm getting to probably is that you can see when it splits in the daughter cell, the plasmids do not replicate in sync with binary fission with the genophore. So now, if this had, when we start with, let's say these are ones that are ability to break down sugar. So this one could break down galactose and lactose. But when we go down to the daughter cell, this one can break down lactose, which is a sugar, and galactose, and use those as food sources as the original cell did. But this one can only use galactose, but is lost the lactose ability because it doesn't have that plasmid. So that's showing you one way how the plasmids can kind of come, or excuse me, in this way showing you how they go. I'll show you how they gain new plasmids. But they can disappear over lineages over time. Okay, binary fission, right? In the nucleoid region, the genophore attaches to the plasma membrane. It then copies itself as the membrane is stretching and elongating. And so it attaches to the membrane and it gets pulled along. Once it's separated, new wall forms in between, pinching in and separating the cell membrane until you have 2 daughter cells. A term that we're going to be getting to when we talk about eukaryotes, haploid. So haploid means there's one single copy of the chromosomes. Here there's only one chromosome, so everything has to be haploid, right? Just remember that term. We'll come back to haploid and diploid later on when we get to eukaryotes, and we'll use multiple. It'll be haploid, diploid, triploid, polyploid. It's a term and a variety of terms we'll be coming back to later. But right now, just remember, all your prokaryotes are haploid. They only have one single copy of the chromosome. Okay, and as I pointed out, plasmids are copied in the cytoplasm and go with the daughter cells, but they copy themselves, replicate themselves independent of binary fission. And so if one daughter cell happens to divide and its cytoplasm doesn't have a plasmid, it disappears. So they can be lost in this process. So this is showing you what I drew for you. So you can see they're trying to show you the origins. Here is where it's starting to replicate. They're not really showing you it attached, but it is attached to the cell membrane, and that's what's pulling it apart. There's where it's attached to the cell membrane. Attached to the cell membrane there. I think it's showing you it's attached there. But it gets pulled apart, and then it breaks apart, and you have 2. So that's binary fission. Binary is the 2. Fission is splitting. Splits in 2. In this process, sometimes when it's making the new, once it's divided, the genophore has separated into 2. Rather than a new cell wall forming on the inside, it can actually, growing in from the inside, it can actually do something a little bit different. So here, if you notice, the cell wall just grew in. But what could have happened, and happens in some instances, is that it could actually form a new cell wall inside the old cell wall, and this is what we would call an endospore. And usually when it's doing this, it's the spore is usually some kind of resistant stage for dispersal. So this spore that's forming might be forming in the soil like in anthrax, which we'll see in a minute. And when winter comes, all of these might die by freezing. But this would survive. So that's the resistant stage. Usually they're being made in advance if a season's coming or a food supply is drying out. Are being used up, water is not available, it's drying out. They'll make a resistant stage. It's basically inert, inactive until the conditions get beneficial, and then it will start growing again. Okay, and so those are called spores. This one's an endospore because, as you can see, it's forming inside. And I want to point out, if you're thinking about the metabolism, it's more than just making a resistant cell wall. It actually has to change the proteins and things so they can, in the membranes and in the cytoplasm, so they can resist drying out or they can resist freezing. So there's more than just making the spore wall in making a spore, but this is showing you what an endospore is, very resistant. One of the better known ones is the endospore formed in a soil bacteria called anthrax, which these endospores and strains of anthrax, which is naturally occurring in soils, have been bred, and they can be very, very toxic, and they're one of these biocide weapons that have been used, unfortunately, in the past. So anyway, that's anthrax, and you can see this very resistant. And you can see it's not just the cell wall, this coat to the outside. It's also changed its cytoplasm and things inside, and even the structure of the cell wall is different. Okay, spoiler alert, when we get to chapter 28, we'll be talking about mitochondria and chloroplasts, and I just want to point out that they divide by binary fission. When we get to eukaryotes later on, we'll be talking about additional ways of cells dividing, and in eukaryotes, they divide by two ways, mitosis and meiosis. Right? We have no mitosis or meiosis with bacteria, so you don't ever want to be using those terms with bacteria. Bacteria are only doing binary fission. And again, they're all haploid. Okay, the next topic I want to take up is also related to the DNA, and it has nothing to do with the replication. It's just how they get their DNA and how does it mix up. And so when we talked about evolution, we talked about the role of mutation making new genes. And of course, mutation is important in these cells as well. So as it's replicating and dividing in binary fission, it might make a mistake when it's duplicating the DNA, and that mistake may be passed down, and that would be a mutation. But here I'm talking about changes in the DNA that's not mutation. We're recombining, meaning mixing up existing DNA. And so there's three ways bacteria can do this. Right? And so the first is called conjugation. And I have it in the middle of the list here. And conjugation is the only thing that's somewhat similar to what happens in sexual reproduction in eukaryotes. And that is that genes get transferred from one organism to another. Right? And conjugation can involve the genophore or plasmids. There are two other ways that DNA gets mixed up. In transformation, what bacteria can do, if you remember these pili are to the outside, they can actually take wild DNA in the environment and absorb it. Usually only bits and fragments of the DNA survive, and that's probably the source of where these plasmids come from. And so you might think, how is there wild DNA in the environment? How does that happen? And that's coming about when cells die. So the bacterial cells, the parasite is attacking something. It's breaking down that host cell. And any of that DNA that survives in the environment, just fragments of it, it can be taken in, and that's by transformation. And this is a natural process, but it's been used in genetic modifications of organisms because they actually can make plasmids in labs and insert them in cells. And so you may hear a lot about transformation in transformed organisms, but it does happen in nature as well. And the third form or way that DNA can get recombined, this is not mutation, this is just mixing up DNA, is in viruses. And right now, of course, we're going through this pandemic with a virus. And so I'll talk a little bit later how viruses work. But basically, in the process, a virus infects a cell and inserts viral DNA. In that process, sometimes the virus picks up a bit of the host DNA, incorporates it into the viral DNA, then it gets transferred to another virus, taking a little bit of the DNA from bacteria A, infecting bacteria B, and bringing the genes across. Right? So that would be transduction. Right? Transformation, external DNA being brought into the organism, making a new plasmid. Right? Conjugation, DNA being exchanged, either genophore or plasmids, between two bacteria, so bacteria to bacteria. Transduction, It's a virus infecting an organism, probably another bacteria, right? Picking up some of the bacteria gene, and then when it infects a new bacteria, transferring the gene from bacteria A over to bacteria B. So that would be transduction. Okay, viruses that attack bacteria we call phages, bacterial phage. And so here we can actually see the phage is inserting genes. And so you can see in here we have A plus and B plus. And the virus picks up the A plus gene into its viral gene when it goes from one cell, infects the other, the A plus gene is inserted, and now this new bacteria has A plus B minus when it originally was A minus B minus. So it's crossing over, right? And so it's coming from bacteria A as A plus, and the virus is the vector transferring that gene. And once it's in here in the genophore, it has this process that you learn about in 130 with eukaryote DNA. But similar DNA can swap out, and so what happens is the A plus gene swaps in and takes over from the A minus gene, and now it's picked up a new trait. So that's transduction, moved across by a virus. Transduction, virus mediated. Conjugation, so here, and I like this because you can see they don't have to be that closely related. This one has all sorts of pili and things going out, Fimbriae probably. But here you notice this is a sex pilus. And what's happening is DNA from one cell is replicated and it moves over and then can get incorporated by that same crossing over process to another bacteria. So in conjugation, here it can be a plasmid, and so replicates itself, moves over, and this has now gained a plasmid. So that's how plasmids can move from one cell to another. Here, it's showing you a particular gene, right? And this, notice they're both starting, they just have one chromosome each. And so it duplicates not the whole chromosome, but it's a bit of it, and then it will do that crossing over again. You don't have to know how that works. You'll learn about that in the cell bio class like 130. But again, this A minus gene, right? We brought over A plus gene, and so it can actually swap over, and now it's got that gene. So it's exchanging genophore to genophore, or gaining a plasmid. Either way, it'd be by conjugation. So conjugation is bacteria to bacteria. There's not a diagram to show you transformation, but basically in transformation, it would simply be taking up as if this weren't here. Let's draw one. So here's our bacteria, and it has its genophore, but no plasmid. But outside here, right, is little loops of DNA. From decomposing cells. And the DNA moves in, so you have a pilus coming out. Right? It gets in contact with the DNA, it moves in, and it's turned into a plasmid. And if it's selected for an evolution, it'll be replicated and passed on into daughter generations of cells. Okay, so those are our three ways that bacteria can change their DNA aside from mutation, right? So there's including mutation, there's 4 ways. But these are now exchanging DNA. A mutation is a change in its own genophore individually as it's replicating. This is getting DNA information from either a dead organism outside, right, via transformation, being transferred from organism to another organism via virus by transduction, or directly exchanging across by conjugation. Conjugation is the only thing that's somewhat similar to what we get in sexual reproduction in eukaryote animals and plants and fungi. Okay, so question, why is antibiotic resistance increasing? And so here you can actually see a chart of some of the more common antibiotics. First, of course, being penicillin. That was the first one discovered. And you can see that within 10 years after its discovery, right, people were becoming quite resistant to it. And there's lots of strains now that are completely resistant to penicillin, and so it's not used nearly as much as it used to for a variety of reasons, but one, of course, is that so many things are resistant to it. And if you look in this amount of time, so many of these ones, very quickly after, some of them just about immediately after they're released, they start becoming resistant. And so we'll talk about why that's taking place. One of them, you can see, it actually took quite a while. And the reason is this colistin actually has lots of really bad side effects. And so it was only ever used when all the other ones weren't effective. And so it wasn't a very strong selection pressure. So if you think what's happening, we have bacteria. So let's say I come to class, and let's say I'm not feeling so well, right? And I have tuberculosis. I turn out to have tuberculosis, which is a bacterial disease, right? Not viral, bacterial, right? But I came to class, I gave a lecture, I had a cough before everyone was wearing masks, and I give it off to somebody else. I've never taken an antibiotic, not even penicillin. I take penicillin and get better. But the person that I coughed on, so that's me, there's student one, right? I cough and give him tuberculosis. I take penicillin, and I get better, right? Unfortunately, this person had already taken penicillin before, right? And you think what happens when you take an antibiotic, it doesn't kill all of the bacteria. So only bacteria that are surviving in her already have this penicillin-resistant plasmid. And so when she gets into the lungs, the bacteria, right, using conjugation, donate, and now that bacteria here, right, has penicillin resistance. So she can't take penicillin. Let's say she takes streptomycin and gets better. But before she did that, she spread it off to someone else. And it has penicillin resistance. But he had taken streptomycin. So in his lungs, all of the bacteria have streptomycin resistance. And so they add on into there the plasmid for streptomycin. So he can't take penicillin. I got better taking penicillin. He can't take erythropoietin because she got better taking erythropoietin. But she had taken penicillin, so they couldn't use that one. So now he can't take erythropoietin, right, penicillin, or streptomycin. And so it keeps spreading until finally you get ones that are built up with multiple antibiotic resistance. And I use the example of tuberculosis because there are strains of tuberculosis that are resistant to ALL, all of the antibiotics. And so there's very little you can do to treat except relying on the immune system. And so that's in part why they've become more resistant. One, it's a very strong selection pressure. We were talking about selection in evolution. The stronger selection pressure, the more it's going to reduce the diversity. And so every time you take an antibiotic, you're selecting for resistant plasmids in your body. And the more people take antibiotics. And so where these build up is where you get lots of transfer in areas with lots of people taking antibiotics. So hospitals, rest homes in particular, build up multiple strains of antibiotic resistance because they're just being used so much and the diseases and the bacteria are moving around and around. Okay, the way that happens, you don't have to know all the details, but antibiotic resistance actually takes two plasmids or two genes on the same plasmid. And anything that gives resistance, we call the R plasmid. And so the R plasmid is the one that actually carry it. You need an F plasmid, which actually lets it be transferred from one bacteria to the next. And so when you get these combinations of F plasmids and R plasmids, then all of a sudden resistance builds up very quick. So you can be donating every time, like I take penicillin. I have lots of bacteria now in my body. The ones that have survived are the ones that have the penicillin resistant R plasmids, right? If tuberculosis, when I get it, already has the F plasmid, it will reach out and be able to take in those R plasmids from outside. So now this bacteria with the F plasmid that can take up other plasmids gets the R plasmid. It gets passed on to the next person, right? They've taken streptomycin, so they have streptomycin-resistant plasmids. It already has the penicillin from me, still has the F plasmid, so it picks up another R-resistant plasmid, and so that's what happens. So the combination of those two plasmids is what makes it the ability to pass on very quickly. Okay, I want to point out that the gene expression is the same. So when we're talking about transformation, it's used a lot in genetic engineering. And so the code, the plasmids, are the same from one bacteria to the other, right? But the genetic code, right, the genes are the same from eukaryotes to prokaryotes. And so that's why you can take a prokaryote gene, like a bacterial gene, and insert it in a plant. And so you may hear about GMO corn. And one of the more common genes that they've added into the corn is actually a bacterial gene from Bacillus thuringiensis, BT. And that has a natural protein that kills off insects that are ingesting it. So when insects eat the corn plant, they actually have this bacterial protein in it that kills the insect. Right? The reason that works is even though it's a bacterial gene and you put it into a plant, they use the same genetic code. Okay, our next one, we're going to be taking up the third part of the chapter, and that will be going over different types of metabolism.